Am I good now? Everything's good? Okay. That's gonna work. Okay, so thanks very much for having me in Philadelphia and having awesome weather. It was beautiful when I got off the plane today. I'm excited to talk to this group. I wanted to make a couple comments. One was thank you to the Peggy Cotter awardees. I have to tell you, in case anybody thought this, just to make sure you know, I am not the funder of these awards. If I could make a big donation like that to ASM, I might, but I don't think I would name it after myself. That actually came, if anybody knows, the year that I was ASM president was a bit of a challenging year. The organization did two things that year. They changed the structure of the governance and also balanced the budget. And when people probably know, if you do things like that, you're going to make some people unhappy. So it was a bit of a rough year. So at the end of my term, the board of directors wanted to create these awards to encourage and support branch members to be able to come to Microbe and support particularly early career members to come. And so I think they kind of felt sorry for me, so they named it after me. But anyway, I'm thrilled that they did, because I certainly really like to encourage younger people to get involved. I was, as Jason mentioned, I was a clinical laboratory med tech for 6 years between undergrad and graduate school. And it was actually while I was an undergrad, my first introduction to the ASM, my supervisor brought me to a Southern California branch meeting and got me excited about the ASM that way. And so that was kind of my introduction to bacteriology through the clinical labs, and then went to grad school. And I appreciated her mentoring right from the start, and that is one of the favorite things that I do is mentor the students that come through my lab. So I was floored when they named that after me. So I'm glad you guys had a good opportunity. And yeah, for everybody in the future, you should apply for those, because ASM, I believe, will support them for a long time. Okay, my second sort of disclaimer is that Jason said I could talk about anything I wanted to today. And so I thought, so at this point in my career, it's kind of fun for me to look back and see how things have changed, and maybe a little bit self-serving about what we've accomplished. And so I'm going to tell you about one project that has been a constant in my lab the whole time. We've taken lots of side roads and tangents and things, but this is the one project that's been the most constant, and really the main reason that I like to talk about it is because it highlights some of the fantastic students that have come through, students and postdocs that have come through the lab and are really the brains behind the project. So I hope you like it. So let's see if I can figure out how to do this. Okay, so this is our project in the lab on Bordetella. Bordetella pertussis is the organism. Let's see if I can control this thing. I really hate this pointer, but it's the only one I have at the moment. Whoa, there we go. Okay, Bordetella pertussis. You can probably just find it yourself. Is the organism that causes whooping cough. It is a strictly human adapted organism that lives only in the human respiratory tract and gets transmitted from host to host. And Bordetella parapertussis also infects only humans and causes an acute disease that is very much like whooping cough, like pertussis in humans. Both of those organisms diverged independently from Bordetella bronchiseptica, or a Bordetella bronchiseptica-like ancestor. Bordetella bronchiseptica has a very broad host range. It infects pretty much all mammals, rabbits, rats, mice, grad students, and postdocs. And it also survives out in the environment, so it's really more of a generalist. Okay, despite the fact that these organisms have different host range and a different propensity, bronchiseptica typically causes a more chronic disease, but all three of these organisms produce a nearly identical set of virulence factors. And so they all produce various virulence factors that people have studied over the years. And the one that I want to tell you about today is the one called filamentous hemagglutinin, or FHA, or as I'm going to tell you next week, we call the big exoprotein FAB. So these are two partner secretion systems that are very widespread amongst gram-negative bacteria. They're a way to get really large proteins across the outer membrane to the surface of the bacterial cell. So the TPSB generically TPS for two-partner secretion. B protein is the outer membrane channel protein, and the TPSA protein is the large protein that gets translocated through the TPSB partner and is the functional protein. And FAB and FAC are the sort of prototypical members of this family of proteins. We know more about them than any other set here. Most of the two-partner secretion systems that have been studied have either proven or postulated roles in virulence, but I'm going to mention one at the end, the CDI systems that are, all right, forget the pointer. CDI systems that are involved in interbacterial competition. Right. We're coming back? Okay. All right, so first I'm going to tell you what FHA does, and then most of what I'm going to tell you about is the work that we've done on understanding the secretion system itself. So FAB mediates adherence. When we first got in the field, so I actually started working on Bordetella to study the regulation system. So there's a regulatory system called BBG, controls expression of all the virulence factors. We learned many things about BBG. It didn't seem to be having quite the role we thought it might initially, so we started looking around for other things to study. And so it turns out that FAB and BBG are right next to each other. So we go, oh, well, we'll study FAB. It's right next to BBG on the chromosome. And so on the top left panel here, you can see the bacteria adhering to, these are L2 cells. So they're sort of a proxy for lung epithelial cells. If we delete the FAB gene, then they don't adhere, but they do stick to the plastic in between the cells. If we delete that regulatory system that controls the expression of the virulence factors, the bacteria only adhere to the plastic. But if we then turn on FAB, then they'll adhere. So FAB is necessary and sufficient for adherence to pretty much any cell type that you put the bacteria on in vitro. And it's also necessary for adherence in vivo. So talented people in my lab developed this assay where we take a mouse, put 500,000 bacteria in 30 microliters down its nose. So you pretty much flood the respiratory tract with bacteria, let it incubate for 30 minutes, and then do bronchoalveolar lavage and see if you can get the bacteria back out. So wild type bacteria, we get about 1,000 bacteria back out, but our BBG minus or our FAB null strain, we get at least 10-fold more. So we assume the reason we can get more out is because they're not adhering in the lungs. And if we do a mixture of wild type and our FAB mutant, they still separate into two. So we think pretty good evidence that FAB mediates adherence also in the respiratory tract. And that's just a different way to show the same data. Okay, so way back, which is probably before some people in this audience were born, early experiments that I did while I was still at UCLA showed that in addition, FAB is absolutely essential for tracheal colonization if we do a more realistic rat model. So here what we do is just put a small number of bacteria in the tip of the nose, 500 here, but we can put 5 bacteria in the tip of a nose and they'll get fully colonized, not so much in the lungs, but nasal cavity, larynx, trachea, and that absolutely requires FAB. So if you just look in the first panel there, our FAB mutant, we did not recover it from the trachea at any time point. Okay, so then we have yet a different model that we can use, and this measures different things. So now we're using mice. And so again, we use the mouse model. We put a lot of bacteria down the nose of the mouse. And what we're essentially doing is looking at whether the bacteria can persist in the lower respiratory tract over time. And we think of this more as a lung inflammation model and a persistence model. Certainly not realistic. Nobody gets infected like that. Okay, so if we look at day 0, our wild type and our mutant strains, we recover, this is, the same data we inoculated just to make sure we delivered the same amount for our wild type and our mutant strains. We recover maybe 3 times 10 to the 5th bacteria from the lungs. At day 3 or 4, what we find is for our wild type bacteria, those numbers have gone up a little bit. But what was surprising for our FAB mutant when we did this experiment, especially the first time, but every time, half the animals, we would recover a similar number of bacteria to wild type, and those animals looked healthy. But the other half of the animals were really sick. And when we had looked at their lungs, they were moribund. We had to sacrifice them, and their lungs showed just massive inflammation. And so, of course, the first time that my students showed me these data, I'm like, well, you messed something up. You need to go do that again. Like, why would half the animals behave one way and half behave the other? But it's completely reproducible at this dose. And after a lot of talking to people and thinking things through, what we realized is that that what's happening is that we're getting sort of a hyperinflammatory response. That is, actually, I should show you more data before I tell you this. If you look at day 11, the wild type numbers are starting to go down. But for the animals infected with the FOBI mutant, that is those that survived at day 3 to 4, they have cleared the infection. And if we look out at day 28, wild type is still not totally cleared, but for the animals infected with the mutant, they've cleared the infection. So, yeah, and then let me also show you these data, which is that if we measure pro-inflammatory cytokines, and I'm just showing 2 here, our FOBI mutant induces a much more robust inflammatory response than the wild type bacteria. And so what we think is happening is that that FOBI is somehow controlling the initial inflammatory response. And without it, you get this more robust response that either controls the infection, so that the ones that are healthy have eliminated the bacteria, or all that inflammation allows it as damage and the bacteria go to higher numbers and it's kind of spirals out of control and the animals die. And so I can tell you that if we just give a little higher dose of bacteria, all the animals will die. If we give a slightly lower dose, all the animals will recover. Okay, so that was maybe a lot of, I'm going to skip that one. A lot of words to tell you that we know that FOBI does 3 things for the bacteria. It allows them to adhere to epithelial cells and other cells in vitro. It somehow suppresses that initial inflammatory response that happens over the first 3 days or so. And it also seems to allow the bacteria to defend themselves against the phagocytic clearance over that next part, so contributing to persistence. All right, so those are the things that we know what it does. So how does it do it? Okay, so FOBI is a really big protein. It's about 370 kilodaltons in its initially synthesized form. It's almost 4,000 amino acids. The about 3,000 or so is the green part up there, which is also the part of the protein that ends up being released into supernatant. And so it got its name because when people early on looked at the protein collected out of supernatants. Sure. Okay, well, hopefully you can see that those look like little filamentous needle-like structures. And so that's why it's called filamentous hemagglutinin. So this is collected out of supernatants. And a little later, Kajava et al. Was able to model this, taking the sequence to model it. And what they predicted, I think this came back. There we go. They predicted this to be primarily a beta helix from the N terminus to almost the C terminus of that part of the protein. And we now know that in fact it is a beta helix because the first 250 amino acids or so have been crystallized, and that's the structure. So that's a beta helix. So it's a long beta helix with sort of a glob at the end. So we just draw it like that. And so, okay, so that's what we knew about the protein. What we knew, there was a model for how it was secreted that was like this. And so this was a lot of really nice work by a woman named Françoise Jacob Dubisson and her group at the Pasteur in Lille. And based on all the data, they came up with this model that made a whole lot of sense, which is that the FOBI protein gets into the periplasm by translocating through SEC, and then it interacts with FOC. And the model is that the N terminus emerges first. It's winding into this beta helix, and that's folding into the beta helix is helping to drive secretion because you may or may not know, but there's no obvious energy source in the periplasm. So for a protein to move to the outside, you need some way for that to happen. And so it's sort of a ratcheted diffusion kind of a mechanism. And then their group also identified this protease called SPHB1. And the model was that once 250 kilodaltons of protein gets to the surface, SPHB1 cleaves, and then FHA is released. So, okay, that was the model at the time. We wanted to ask the question, if this is an adhesion, why are the bacteria releasing it, right? Why are you letting go of the thing that you are needing to hold on with? And so with Bronchoseptica and animal models, we have nice animal models. We can study lots of different aspects of the infection. We wanted to ask the question, is that actually biologically relevant? Is it important to release this protein that is apparently an adhesion? And of course, an easy way to do that is just delete the gene encoding SPHB1 based on a model. And then and then infect animals and see what happens. So Joseph Mazur joined my lab to do just that. And what he found is that, in fact, SPHB1 is not the only thing responsible for the cleavage. So on the left here, I'm showing Western blots. And this top band, there, that's FAB, that's the full-length protein. Down here, what we see are several proteins of about 250 kilodaltons. And in the supernatant, you only see the smaller proteins. That, what we call the prodomain at the end of the protein, we never see it by itself. It seems to get degraded. It's not like it's just cut between the green and the blue there. Okay, but what Joseph found is that if you delete SPHB1, you still get cleavage, you still get a 250 kilodalton protein, and it still gets into the supernatants. And he found that the same was true. You get these different forms of it that we give different names, doesn't really matter. But the same was also true for pertussis. And so we said, so now we can't ask the question, we can't just delete SPHB1 and say what happens when you don't release the protein, because that won't work. And so that was when we decided, well, I guess we don't know enough as much about the secretion process as we thought we did. So then we became members of the protein secretion community, had to do some experiments of our own. And so Joseph works really hard on this for like 3 years. He's doing all these experiments, working really hard, and beautiful data, and none of it made any sense. And it was just like, oh, you know, how can this be? And it wasn't that the experiments weren't working, it was just that they just didn't fit the model. And then he finally did this experiment, and you know, everything made sense. And I remember the day, I remember sitting down, my hands were literally shaking from what he did. And I'm not going to show you that experiment because it's kind of more complicated, harder for me to describe. This one is both simpler and more aesthetically pleasing, in my opinion. And so what he did here is he put an HA tag at the N-terminus, and then we have an antibody that recognizes the middle of that beta helical region, and he did these dot plot analyses. So we can look at whole cell lysates or supernatants and basically look and see which part of the protein is accessible to the antibodies. And so what you see is if he looks at whole cell lysates here, only the green antibody is recognizing it. But in supernatants, the red antibody that recognizes that HA tag is recognizing it. So let's see if that's supposed to show up. Okay, so only the secreted protein is getting detected by the antibody that recognizes the N-terminus. But if instead he puts a his tag at what should be the C-terminus, that actually gets recognized by both on the whole cells and in the supernatants. So what that's telling us is that model was upside down. And it doesn't, right? These data here don't fit that model. Instead, it works like this. And so what seems to happen is that the N-terminus of FAB stays associated with FAC, and you build this beta helix pulling out more and more protein until finally SPHB1 cleaves at this junction, and then the protein looks like this on the surface. So this told us a lot of things. It told us, for one thing, if you're wondering which part of the protein might actually be interacting with a host cell, it's not the N-terminus like the first model, which told us it must be that C-terminal part of what we sometimes call the mature protein, or the MCD, is that green region. Okay, and then you know you have this what we call the prodomain region that gets degraded in the periplasm. Okay, so that was a surprise for us. That was a big deal. And then Chris Noel joined my lab, starting in Santa Barbara, moved with me to North Carolina, and Chris got very interested in that prodomain. The prodomain is 1,200 amino acids. It's a respectable sized protein on its own. And he thought it must be doing something. And so he started looking at it. And the first thing that he found is that this region that's near the N-terminus of the prodomain, that he calls the PNT, the prodomain N-terminus. Well, hopefully you can see that. What he showed is if you delete that region, the prodomain just goes out. Right, it just zips right out to the surface. And so the PNT seems to be something that stops secretion of the rest of the protein. Okay, so then he looked at the very C-terminus, which he cleverly named the ECT for the extreme C-terminus. And it turns out those last 98 amino acids are identical for every FHA protein that's been sequenced. So we have a lot of sequence for Bordetell strands. Figured if it's going to be identical, it must be important. I've never had so much trouble with my computer. So sorry. It's back. Okay. Maybe I just need to stand on this side. Maybe I'll try that. I don't know how that could do it, but anyway. So now we have AlphaFold too. That's what AlphaFold predicts its structure to be like, or whatever that's worth. Okay, and then there's a region right upstream of there that, for obvious reasons, we call the proline-rich region, and AlphaFold predicts it to look like that. It's not very helpful. And so anyway, so Chris got interested in what do these parts of the protein do? And so he made strains that produce proteins with either the one or both of those regions at the end deleted. And the main point here that he saw is that if we have a strain that does not have the ECT, then you don't see the full-length FOB protein. You only see that process protein. So that one, and it doesn't matter whether you have SPHB1 or not. So what that suggests is that very C-terminus of the protein is somehow inhibiting a protease that would degrade the whole protein. And that also suggests then that this is a regulated process and that really that regulated degradation could be important. All right, so then Jeff Melvin joined the lab and he said, okay, you can do that in vitro, but does it mean anything in vivo? What happens if you infect animals with these strains? And so what he showed, so now this is similar to data what I've already shown you, but now I'm showing you as sort of these scatter plots. Our FHAB mutant, at day 3, we get those 2 different populations, and at day 11, the ones that have survived have cleared the infection. With the 2 mutants, I think I missed something. Let's see, there we go. Okay, if we delete the ECT or the PRR at the end of FOB, what you see is that we don't get that, what we call this sort of bimodal thing happening here, but they are cleared as fast as the Delta FOB strain by day 11. Okay, so the end of the protein is important during infection. And I'm just going to skip that because I know we're probably running a little late on time. So the point here is that these 2 domains in the protein are playing an important role during infection, but this whole protein just seems to get degraded, right? And so I think typically we think of like, if you have a protein and it gets processed and this happens, and then you can detect this, you think that's the final form, that must be the functional form. But what Jeff Melvin said is that, I think what my data are telling us is that this final, process form is not the functional form. The full-length protein must be important during infection, because if we make mutations in the protein, which is only present on the full-length protein, we're getting a dramatic effect in vivo. And so it really sort of changed our view of this project, and it made us think that not only is the full-length protein important, but probably that regulated degradation of the protein is important. Okay, so we think that perhaps some signal happens on the outside when FOB binds its receptor, whatever that is, and that then maybe causes processing. That was our hypothesis. And we thought maybe we could start to understand this if we could identify the protease. Okay, so we don't know what the signal is, but we also didn't know what the protease was. So Zach Nash joined my lab. I asked Zach for a picture, and this is the picture that it gave. So I do like it. And so what Zach did was identify the protease. And I got to tell you, he just guessed. He happened to look. So how many proteases are predicted to be in the periplasm? And there was this one that is supposed to be a member of the family of C-terminal proteases. And he goes, well, I think this thing is cleaving out the C-terminus. So he knocked it out and it was it. And so, so this protein has the catalytic dyad in here. It's got this PDC domain, a signal sequence. It gets in the periplasm. And so this is basically the evidence for this, which is, so I guess if we just are patient, it'll come back. Ooh, okay. So now we get 2 colors. So yellow is being detected by both antibodies, the one that detects what we call the MCD, and one is this HA tag that we put near the end of the protein. and green is the processed protein. So I told you already that if you delete the extreme C terminus, you get hyperprocessing and you only see this 250 kilodalton protein. So Zach deleted that gene called CTPA, and you can still see the full-length protein. The prediction would be you would only see the full-length protein if this was required for degrading the protein. But we saw a different size protein. And so that says that CTPA is involved in processing, but it's not the only protease. Of course, it's not going to be that simple. And we get this 275 kilodalton protein. But another thing that was important from this, it was actually, so I already told you this, but I didn't show you the data, that we never see the protein on its own. So if we try to look, so this red antibody here is staining protein fragments. Well, staining any protein that has the protein, and we never see that in our whole cell lysates. But if we delete that protease, that CTPA protease, we see bands corresponding to having, it's apparently cutting it at multiple places. We don't know where. Okay. I'm going to skip that slide too, because I don't think you needed to understand the story. So, okay, so this led to this model. So FAB is made as this full-length protein. We think that some signal gets goes across the membrane and tells the extreme C terminus maybe to change conformation. So another protease can cleave off that very C terminus, and then the CTPA protein degrades the rest of the protein. Where's the loose connection? Okay, and then we get this protein that we call FHA prime, and then maybe SPHB1 cleaves it, and you get the final protein. So that's our model. And then, of course, the question is sort of, well, here's the question that Richard asked. First, the next question he asked was, what's that other protease, right? So if we identify the other protease, maybe we'll understand things better. So this is just to show you, some people like to look at this linearly. So we have our full-length protein, then there's an initiating protease that cleaves off the end, and then CTPA degrades the protein, and then SPHB1 cleaves, and then you get FAB. And so Rich took all of the genes that are predicted to encode proteases that would get in the periplasm, rather than just guessing one and being right like Zach. He tried all of them, and he found one. And the one that he found is DEGP. So you may know DEGP can be a chaperone and a protease. And so, in fact, if he deletes DEGP, we pretty much only see full-length FAB protein. And if he just makes that S to A substitution, so it'll be catalytically inactive, we also see only the full-length FAB protein, and he can complement. So DEGP seems to be the initiating protease. And so we have DEGP cleaving off the C terminus and CTPA degrading the protein, and then finally SPHB1 cleaving, and then FHA is released. Okay, so here is the question. Why is this so complicated? Right? What is the point of that? Like, you get different proteases chewing this protein down to a 250 kilodalton protein that then just gets released from the cell. So what's going on? So to tell you about that, I have to tell you about a different virulence factor called adenylate cyclase toxin. So this is a big complicated protein in itself, and I'm going to try to keep it simple. It has two domains that are important. At the N terminus, it has the adenylate cyclase domain. And what that does when it gets inside eukaryotic cells is it binds calmodulin and then produces huge amounts of cyclic AMP, which of course is going to screw up signaling in the host cell. The whole C-terminal part is a member of this RTX hemolysin family of proteins. So that stands for repeats and toxins. And there's a pore-forming domain, and in fact, this protein can form pores and membranes, and so the bacteria are hemolytic if they produce this protein. But it also has a binding site for CD11B down here in the middle. So CD11B is a receptor that's on phagocytic cells. So macrophages and neutrophils in particular, in dendritic cells. Okay, so that's what you need to know. Catalytic domain at one end, phagocyte binding at the other in the middle, and it gets secreted by a type 1 secretion system. It turns out, I'm going to tell you what it does. So if you delete SIA, the bacteria get cleared faster than if they have SIA. Okay, so At 72 hours there, the SIA deletion mutant and the catalytically inactive SIA strain are a log or two lower than wild type, so they're contributing to persistence in the lower respiratory tract. And this is an experiment from a very long time ago, but it's very informative, which is if we infect skid beige mice, so skid beige mice have no adaptive immune system, no B cells, no T cells. So they're reliant on their innate immune system to be protected. Wild type Bronchoseptica kills skid beige mice because you need your adaptive immune system to control the infection. But the delta SIA mutant does not kill the skid beige mice. So what that tells us is the innate immune system is sufficient to control a SIA mutant. So the way we think about it is this: if the wild type bacteria, they can overcome the innate immune response with ACT, but then your adaptive immune response, the T cells and B cells, control the infection. And so it doesn't matter whether you have SIA or not, you've got the immune system you need to protect you. But in skid beige mice that are being T cell deficient, wild type can still overcome the innate immune response, and then it ends up killing the mice. But for the SIA mutant, it's still, they can't even overcome that first step. So the main point of all that is just that ACT is required to resist the clearance by the phagocytic cells that come in, which I told you FAB also does that, right? It seems our data suggests that ACT and FAB might be working together to resist the clearance by the innate immune system in particular. So some collaborators of ours actually found out that FAB and ACT bind to each other on the surface of the bacteria. And we collaborated with them, Rich and Zach did. And so this is showing this by a dot plot. So we're just looking at antibodies that recognize either ACT or FAB on the surface of the bacteria. If we have wild type bacteria, they have both proteins on their surface. In our Delta FAB strain, there's no ACT on the surface of those bacteria that we can detect by dot plot. So without FAB, ACT just goes right into the supernatants. It doesn't stay on the bacterial surface. Without SIA, you still have FAB. Okay, and this is just to show you that in fact the proteins are there in that FAB mutant. It's just a matter of where they're localized. Okay, so then Zach did this experiment that is one of those experiments where students say, yeah, I want to do this experiment, and I'm like, I don't understand why you want to do that, but go ahead. And what he did was he treated the bacteria with proteinase K to get rid of pretty much all the surface protein. And then he looked over time at FAB and ACT getting back on the surface. And he did this in a wild type and a FAB mutant. And so on the left, our dot plot showing that if you treat with proteinase K, you can get rid of all the ACT, but by 30 minutes, you can see it on the surface again. And if we look at FAB on the right, you can never quite get rid of all of it with proteinase K because that beta helical structure is very kind of resistant to the proteinase K treatment, but you can knock it down and then it comes back. But what was mostly interesting was actually when he looked in supernatants. And so what he saw here is that with the FAB mutant, you see ACT in supernatants much faster here than for wild type. For the wild type bacteria, you don't see ACT on the surface or in the supernatants until 2-3 hours later. So what this experiment showed us is that the bacteria first produce FAB, and then we also know that ACT lags a little behind FAB, but then they produce ACT and it sticks immediately to that FAB, and it doesn't go into the supernatants as long as you have FAB. And it doesn't go into the supernatants unless either we think all the FAB is bound to ACT, or maybe they go off as a complex, which is what that was supposed to show. Okay, and then our collaborators and showed exactly which parts of ACT bind to which parts of FAB. So the catalytic domain binds to the mature C-terminal part of FAB. Okay, so that brings us to this model. So we think FAB is first produced as this long protein with this structure on the surface. That ACT, which gets secreted by a different secretion system, then binds to it, and it binds to that big distal part of the protein, and it's stuck there. I should tell you this. It turns out that if you add adenylate cyclase toxin to epithelial cells, you really don't see any intoxication. And I told you that ACT it can bind the receptor that's on phagocytic cells. So if you put ACT on phagocytic cells, it intoxicates them, but not very efficiently. But if you let the bacteria deliver it to those phagocytic cells, it's very efficient. And so what we put all that together is to think, if the bacteria, what I'm trying to show across the top now, if FAB binds its receptor, which may be an epithelial cell or any other kind of cell, then nothing happens. If it's a phagocytic cell with CR3, then ACT binds to that cell. And what we think maybe that is the signal that gets transduced across the membrane into the periplasm and tells DEGP to cut off the last little bit of the protein, and then CTBA cleaves, and it chews halfway through that PNT, which is the part that keeps the rest from going up. And when that happens, the rest of the prodomain, that little bit that's left, sort of slides out, and now ACT can get delivered to the host cells and intoxicate it. And then once that's been done, when that's finished, you don't need FAB anymore because it's delivered that toxin to the host cells. And so SPHB1 cleaves what's left and the whole thing gets released. So why? Let me think of what I was I'm trying to make sure that I stay on time a little bit. I'll tell you this experiment. Okay, so we can run bacteria on phagocytic cells like macrophages, and we can measure cyclic AMP, and we will get wild type induces a lot of cyclic AMP. A delta cyanide, no induction, as you might expect. Our delta FAB strain, so we actually spin the bacteria down to get them, make sure that they're all sort of at the same time interacting with the host cells. So it's really not too surprising to us that some ACT would get secreted and go into the host cell, even in the absence of FAB. But what was a really important experiment for us is if we delete DEGP, we don't see any intoxication. And we think it's because without DEGP, you can't cleave the prodomain, so then you can't sort of loosen up what's out there and get ACT delivered to the host cells. So that's one piece of evidence for this model that we have, that in fact they're working together, and regulated degradation of the prodomain is important to deliver that toxin to the host cells. Okay, then, so that's our model. Zach did one more experiment. I love this experiment. He was so clever. He could take an ACT minus bacterium. So this one cannot produce any ACT on its own, but it's got FAB on the surface. And he can add to it supernatant from a bacterium that does produce ACT. And now that ACT will stick to FAB on the surface. And so the purpose for this experiment was to really ask, can the surface-associated ACT get delivered to host cells, not the newly produced ACT, right? Because there's no newly produced ACT here. So he takes those guys and he incubates them with phagocytic cells, measures cyclic AMP. It's not a huge amount, but we're not surprised at that because they're not making any new ACT. It's just whatever we could coat the bacteria with. But you did induce cyclic AMP in that experiment in the host cells. Okay, so why would the bacteria do it this way? So Bordetella likes to colonize the ciliated epithelium of the respiratory tract, particularly the nasopharynx and the trachea. And so they get in there and they're gonna bind to the ciliated cells and start growing. And they don't want to intoxicate those cells, as far as we can think, it would be to their advantage because that's a nice sort of place for them to live. But of course, they're going to induce an innate immune response. And so we think that by ACT binding to FAB and using that as a specific delivery system just for phagocytic cells, that's a way to defend against the phagocytic cells with doing sort of as little damage as possible to the epithelial cells that they like to colonize. So that's our model for that. Okay, and then just in the last minute or two, I'm going to tell you that I think maybe all two-partner secretion systems are contact-dependent cell surface, receptor binding specific toxin delivery systems. So I mentioned earlier that there are these proteins called CDI systems that some bacteria have. And so it's an analogous protein to FAB, but instead of binding to a host cell, they bind to other bacteria. And the toxin they deliver is their actual C-terminus of the protein. And Chris Hayes showed this for E. coli, and or at least has a model that looks a lot like ours, I think, that the CDIA protein is in this conformation and the toxin remains in here. And when this receptor binding domain binds a target bacterium, instead of degrading the prodomain, it resumes secretion out so that it can intoxicate the target cell. And so it's the same concept, same kind of protein, but instead of delivering a different toxin, it's actually the C terminus of that protein, which is pretty cool. And then I just want to, so those are those guys. So this protein, IBPA from Histophilus somni, is another TPSA protein with a toxin at its C terminus that it delivers to host cells. Okay, and so we think that there's some good examples that this whole sort of, it's not all of the two-carbon secretion family proteins, but it's most of them. There's like one other class that's different. That all of these might actually be a way for the bacteria to do contact-dependent signaling, knowing when they bound when they're interacting with their target cell, and then allowing them to then deliver a toxin, either the end of the same protein or a different protein into those cells that either mediate bacterial host interactions or bacteria interactions. So we have a lot more work to do to see if this is going to be true at all or generally. Anyway, for me, it's a kind of satisfying to look back at, you know, what the model was when we got into the field and how far we've come because thankfully of the work of lots of very talented people that have come to my lab, and I'm very appreciative of it. So I think I mentioned pretty much all of these people. I don't think I mentioned Steve and Carol during the talk. They did a lot of the early animal work with our FOB mutants and ACT mutants. I'm very appreciative of funding mostly from the NIH. This is the current, well, not even the current lab anymore because we're evolving fast, but I wanted to show sort of the most, trying to think if anybody up there that works on this that is not mentioned here, and I think not. Anyway, that's where we've come. Mostly I want to thank you for your attention. I'd be happy to answer any questions.
What are the themes? What happens if you just add supernatant, which has the virulent similarity with the toxin to the cells? Do you really need this contact from the bacterium, or because FHA gets cleaved at the end,
Right.
So would that be enough with the secreted toxin to bind now to the FA, they're both together, binding on the surface and then just being released, and now that actually is also very high.
Yeah, we have not done that experiment. I'm not confident that if you try to get FHA from the supernatant and ACT from the supernatant and mix them together, I don't know if they would bind.
Well, they might bind before, so if you
yeah, so if you just collected supernatant that in
which
FHA and ACT that are still sort of stuck to each other, could you add that? I don't know. Maybe we'll try it.
Okay.
That was really fun. How long does the FHA domain to interact with the host cell? Has anyone sort of cut it down and said, okay, what is the minimum there? Because that's a big protein. So what are
It is. So now you're getting into what I debated to adding a bunch of stuff at the end here, because as I showed you, we draw this as like a big glob. I mean, a big, sort of a protein with a glob at the end. I'm going to just go back a little bit to here. So now that we have, with AlphaFold 2, we can start to get some super interesting predicted structures for the different parts of the protein. And so like some of these structures in here are really interesting looking, but related to what you just asked, it turns out that for the CDI systems, so as exemplified here, the length of the beta helix and the length of that long part that comes down have to match. And so you'll have some strains that for some of the systems, you might be missing 200 amino acids of the beta helix. and you're missing 20 amino acids of that strong part from of that long part. But that's about the same length, right? Because the beta helix is super twisted up. And in fact, we're doing those experiments now to mismatch them. We already have some evidence that you can't. For FOB, it's a little different because in fact, for pertussis, which is the MCD region is identical to the one in bronchoception, but it also has a little shorter beta helix. So everything's not the same for all these systems, and but yeah, there's some really interesting things like how long, why does this have to be so long? Is it so that the end is sticking far enough out over LPS or whatever? Or is it really just so that you can pull out enough more protein? Because that's the other thing that beta helix is doing, is that as it grows, it's pulling more protein out. There's so many interesting like biophysics problems here too, right?
Yep.
Yeah.
I have a quick time to remember. The biphasic response in mice, some died, some survived. Was that explained?
Well, the best that we know is that what's happening is that because it's this hyper-inflammatory response, that it's really just, so the way this got explained to me, because I thought, how could this means that this doses the LD50 for the FOB mutant, right? Half the mice are dying.
But it's so clean, it's such a big difference.
Yeah, what does that mean? What does that mean? So I talked to this physician one day, and he goes, well, okay, if you get pneumonia, you either recover or you die, right? And you don't just like stay sick forever. Like we never did LD50 experiments. I imagine all the animals are sick and half of them are dying. But in fact, that's not it. They're either recovering or they're dying, right? And so I think that pneumonia that they're experiencing with that mutant is just sort of on this knife edge. And we know it's a hyper-inflammatory environment, and it's sort of by chance who wins that particular one. Yeah.
Which was stochastic, it's just.
It is a bit stochastic. One of the things that we really learned from that is that, you know, sometimes when people give you a bar graph and, you know, maybe they got error bars or whatever. And when my student showed me the data instead of a bar graph showing me this, I was like, those are two different groups, right? This wasn't a spread. This is two different groups, and it really changed the way we think about evaluating the data by realizing that these are two different groups. And of course, first we thought maybe some are bacteria mutated or whatever, but with enough, you know, we realized, no, this is what these guys do. Yeah. Thanks.
Super cool talk. My question is a little bit more sort of conjecture based off of your last slide with all the different systems across the bacteria. Is that CDI primarily targets like other prokaryotes, and then FHA primarily targets eukaryotes?
Yeah.
So are you able to use any part of the sequence to kind of predict if a newly discovered or characterized protein is going to primarily target prokaryotes and eukaryotes? And the second part kind of related to that is, you know, prokaryotes have other kind of contact dependent systems like type 6 secretion systems. Is there sort of like crosstalk between CDI and type 6 and any other systems bacteria use to sense their environment essentially?
Yeah. So your first question, we are super excited about that question. What is the specificity? And it turns out for some of the CDI systems, I really wish I had put this. Looking for a whiteboard. The way the structure is predicted to look, and then we can compare that to who infects who. We can sort of start to pick apart. This is the part that's involved in specificity, and so we can start to do some swaps and see that. And then the other sort of cool idea is, can we swap between? One of the things we would love to do is be able to put the bacterial specificity to FOB. So we could take our Bordetella and another bacteria and start to look at, because to be able to look at what's happening when we interact our bacteria with a eukaryotic cell is more difficult, right? And then, so my postdoc is going nuts with all these ideas about, like, you know, if you can not only do the specificity, but deliver a toxin, then you can deliver toxins to cancer cells or something, right? With the right formation that you have. Yeah, so there's a lot to do with regard to that. What was the second part?
Interaction with like type 6 secretion systems.
Oh, yeah, so a lot of bacteria that have CDI systems also have type 6 secretion systems. So one of the differences is that type 6 secretion systems tend to be sort of, I shouldn't say indiscriminate, but it's like any other bacteria that's there, I'm going to try to kill it. The CDI systems are actually incredibly specific. And so that's the other thing that we're not studying CDI so much in my lab anymore, but what we were puzzled with is these are ways of killing other bacteria, so why would you kill your siblings, right? So there's almost so much specificity that the only other bacterium that would be susceptible is like a sibling. And so what's going on there? And we don't understand it. I mean, they're not siblings because they don't have the right, so they need an immunity protein to protect them if they don't know when they die. So we don't really understand that yet. But the two, they seem to work a little bit differently that way, but there are a lot of bacteria that have both, that have type 6 secretion systems and CDI systems, and whether they use them at the same time or in the same context, I don't think anybody knows yet. Thanks.
So I want to go back to that lipase response. So the fact that the EC domain deletion doesn't have that, does that mean if the protein is highly processed, you can still deliver that ATC to the green cells?
So I'm glad you asked that because I know I went pretty fast through that part. So we think, so in vitro, without the ECT, we see that rapid degradation of the protein. I'm not sure that's what's happening in vivo, because we don't know what the signal is that actually, we kind of think that when we see in vitro the protein get degraded, that's kind of an aberration, an artifact of growing them in vitro, because they're not binding their targets at all, right? But they're bumping into the glass and each other and all that. And so we don't really know so much about that part of it. But by not seeing the hyper-inflammatory response initially, we think that what we're actually seeing so we think FOBB maybe is doing two things, whether they're totally related or just overlapping. One is suppressing that initial inflammatory response. And it seems that our PRR mutants and our ECT mutants are capable of doing that, because we don't see the hyper-inflammatory response. But they're defective for sort of the second phase, the defense against the phagocytic cells that get recruited. And so we don't know anything really about how this control of the initial inflammatory response works. But they seem to be either related or two separate things that FOBB can do, and the protein is only important or most important for the second one.
And for the third thing, that's the EC domain region, that's the bacteria adhere to the cells.
Yeah, they still adhere. Oh, and I didn't even show you this. They still adhere in vivo, even in the animal, right? So they're only defective for that defense against phagocytic cells, and not defective for adherence either in vitro or in vivo.
All right, running a little late, which is great. Good conversation, good questions, great thought. So let's thank our speaker again for.
Thank you.
Sorry for the chaos.
I'm sorry for my computer being